Bismillahirrahmanirrahim. Students, today we will discuss graphic life cycle and diagrammatic life cycle of Pinus. First, we discuss the diagrammatic life cycle of Pinus. Diagrammatic life cycle is that cycle in which the life cycle is represented by the diagrams. All stages are represented by the diagrams. As we know that Pinus plant body is sporophyte and it is heterosporous. On the plant, female cones and male cones are produced. Female cones are formed by the arrangement of the megasporophylls, and on these megasporophylls, this is one diagram of the megasporophyll. On these megasporophylls, megasporangium are produced. On one megasporophyll, two megasporangium are produced, which are known as ovules. And in the nucellus part of the ovule, megaspore mother cell is produced. And then in the female cones, which are also formed by the arrangement of the microsporophylls, and on the lower side of the microsporophylls, two microsporangia are produced, and in these microsporangia, microspore mother cells are produced. Then reduction division occurs in this megaspore mother cell and in the microspore mother cells, and as a result of reduction division in megaspore mother cells, 4 megaspores are produced, out of which 3 are degenerated and only 1 survives, and it acts as the functional megaspore. In microspore mother cells, after reduction division, tetrads of 4 microspores are produced, and then these microspores, still when these are present in the microsporangium, they start to germinate, and at 4 cell stage, these germinated microspores, which have wings on these microspores, which have wings, and these germinated microspores with wings are known as the pollen grains. Functional megaspore mother cell, it germinates, and then after germination, free nuclear division occurs in this megaspore mother cell nucleus, and after free nuclear division, cellular tissue is formed by the wall formation from the outer side towards the inner side, and then as a result of this cellular tissue is then known as the female gametophyte, or it's also known as the embryo sac. And in this microspore, which is germinated and known as the pollen grain, this is the male gametophyte. Now, in the female gametophyte, in the upper part of it, female gametophyte or embryo sac, archegonial initial cells are distinguished, and from each archegonial initial cell, one archegonium is produced, and the structure of archegonium is that it consists of neck cells and one ventral canal cell and one egg cell is present in the archegonium. And in the female gametophyte of different species, 2 to 6 archegonia may be developed. This pollen grain, it is present in the microsporangium, and then these microsporangia, they release these pollen grains from the microsporangia, and then further development of this pollen grain, it occurs in the pollen grain when it reaches after pollination to the female gametophyte. When after reduction division occurs and haploid megaspore megaspore and microspore are produced, this is the start of the gametophyte phase, gametophyte stage, which is haploid stage. And then when pollination occurs, these pollen grains, they are carried to the female gametophyte, entered through the micropylar region, then nucellus, and then when these fall on these nucellus tissue of the ovule, and then they reach to pollen chamber, and then when they fall here, germination, further development of these pollen grain occurs, and in this part the pollen tube arises. This is the pollen tube, which the formation of pollen tube occurs here, and this pollen tube, it reaches to the female gametophyte, and then it enters into the pollen tube. It releases the male sperms into the archegonia, and then in this archegonium, where egg cell is present, by the fusion of the male gamete with the egg, a zygote diploid cell is formed, and here this is the end of the gametophyte stage. And then after fertilization, on fertilization, this is the start of the sporophytic phase. After fertilization, when zygote is formed, then germination in zygote occurs. This zygote is germinated, and then division occurs in the zygotic cell, and as a result of the germination of this, embryo is formed. And this embryo, when it is formed in the Pinus species, polyembryony is present, cleavage polyembryony, where more than one embryo are developed, and then usually one embryo develops and all the others, they are degenerated or abort. This is the embryo, and embryo, they have suspensor cells on their backsides, and these embryos, they the external structure of the embryo is like this, which is elongated structure, and then at the side of this, whorl of cotyledons are present, and then in the axils of these cotyledons, the shoot apex arises. यहाँ से shoot apex जो है वो arise होता है. This is the internal structure of the embryo. This in this embryo, some changes occur, and as a result of these changes in these embryo, it is converted into the seed. This is the outermost seed coat or testa or tegmen, which is formed by the integument layer, by the changes which occur in the integument layer. And this is the micropylar region. Here the suspensor, remains of suspensor cells are present, and then here hypocotyle is present, here radical is present, जहाँ से root arise होगी, और ये जो है वो shoot apex है, जहाँ ये cotyledons का जो है whorl है, और इसके बीच में जो है वो shoot apex arise होगा. This is the internal structure of the embryo. ये last lecture में seed की structure है, जो last lecture में हम detail में पढ़ चुके हैं. Then this seed, it germinates when it falls on the suitable place for its growth, and on germination, the hypocotyle region, it has radicle, and this radicle, it grows to form the root, and this root enters into the soil, and from the epicotyle plumule arises, और ये जो cotyledons हैं, इनके बीच में जो है सेंटर में जो है वो shoot apex rise होता है. And in this way, after germination of the seed, again the seedling stage is produced, and this seedling stage जो है ये sporophyte plant body जो है उसकी जो है वो उसको produce करती है. And then in the axils of these cotyledon, जहाँ से शूट अराइज होता है, वहाँ पर dwarf shoot जो है स्पर अराइज होना शुरू हो जाते हैं. And in this way, the seedling is developed into the sporophytic plant body, and at last the life cycle of the स्पोरोफाइट इज कंप्लीटेड। तो पाइनस प्लांट जो है इसमें भी जो है वो रेगुलर अल्टरनेशन ऑफ जनरेशन पाई जाती है, जिसमें गेमेटोफाइट और स्पोरोफाइटिक स्टेजेस जो है वो अल्टरनेट करती हैं। डोमिनेंट फेज जो है वो स्पोरोफाइट है और गेमेटोफाइट फेज जो है वो शॉर्ट लिव्ड है। नाउ नेक्स्ट वी सी द graphic life cycle of the Pinus. Graphic life cycle is the life cycle in which the stages of the life cycle, these are represented by the names. Pinus plant body हेट्रोस्पोरस, इसके ऊपर फीमेल कोन प्रोड्यूस होती है, मेल कोन प्रोड्यूस होती है। मेल फीमेल कोन में मेगास्पोरेंजियम जिसको ओव्यूल कहते हैं प्रोड्यूस होते हैं, और मेल कोन में जो है वो माइक्रो स्पोरोफिल जो है उनके अंदर माइक्रोस्पोरेंजिया प्रोड्यूस होते हैं। और इस तरह से मेगास्पोरेंजियम में मेगास्पोर मदर सेल, ये हम पीछे डिटेल में जो है वो पढ़ चुके हैं डायग्रामेटिक लाइफ साइकिल में। और माइक्रोस्पोरेंजियम में माइक्रोस्पोर मदर सेल प्रोड्यूस होते हैं। देन रिडक्शन डिवीजन अकर्स इन दिस, and this is the here end of the sporophytic phase, and after reduction division start of the gametophyte phase. From megaspore mother cell megaspores are produced, 4, and one survives, which on germination produces the female gametophyte or embryo sac. And microspore mother cells जो है वो आफ्टर रिडक्शन डिवीजन प्रोड्यूस द टेट्राएड्स ऑफ माइक्रोस्पोर्स, और माइक्रोस्पोर जो है वो जर्मिनेट करके जो है वो मेल गैमीटोफाइट जो है वो प्रोड्यूस करते हैं, और मेल गैमीटोफाइट जो है वो उसके अंदर मेल गैमीट्स प्रोड्यूस होते हैं। सिमिलरली इन द एम्ब्रियो सैक और फीमेल गैमीटोफाइट जो है वो उसमें आर्चिगोनिया प्रोड्यूस होते हैं, और आर्चिगोनिया जो है उसमें एग प्रोड्यूस होते हैं। और हमने पढ़ा कि आर्चिगोनिया जो है वो 2 to 6 आर्चिगोनिया में बी प्रोड्यूस्ड इन द डिफरेंट स्पीशीज, एंड ईच आर्चिगोनियम जो है इट कंटेन्स 1 egg cell, and then when male gametes after pollination these reach to the archegonium egg cell, fertilization occurs, and as a result of this fertilization, here it is the end of the gametophyte phase and start of the sporophyte phase. After fertilization, the zygote which is produced, it is also known as the oospore, and on germination it produces the embryo, and embryo is then converted after some changes in the ovule, it is converted into the seed, and seed on germination again produces the sporophyte plant body of the पाइनस। तो स्टूडेंट्स हमने पाइनस की टाइप जो है वो कंप्लीट कर ली है। पहले इसका मॉर्फोलॉजी और जो है एनाटॉमी और इसकी मॉर्फोलॉजी लाइफ साइकिल हमने डिटेल में पढ़ा, और आज हमने इसका लाइफ साइकिल, ग्राफिक लाइफ साइकिल और डायग्रामेटिक लाइफ साइकिल डिटेल में पढ़ लिया है।